Welcome back. We're going to cover Chapter 7 today, movement. The control of movement. Think about your day so far. Maybe you got out of bed, walked to the kitchen, made coffee, or sent a quick text. Every one of those actions, from stretching your arms to tapping your phone screen, is made possible by a highly coordinated system of muscles, neurons, and feedback loops. In this lecture, we're going to explore how movement is controlled from the basic building blocks of muscle fibers to the spinal reflexes that keep us safe to the higher brain regions that orchestrate voluntary action. Let's start with the basics. Muscles. Movement begins here. There are three major types of muscles in the human body. Skeletal muscles are the ones under your voluntary control. They're striated, meaning under the microscope, they look striped and they let you run, jump, write, or even smile. Smooth muscles handle automatic processes like moving food through your digestive tract. Cardiac muscle, of course, powers the heart, and it's specialized to keep beating rhythmically. How do skeletal muscles work? They don't act alone. Each muscle fiber is connected to a motor neuron. When that neuron fires, the muscle fiber contracts. Together, one motor neuron and all the muscle fibers it activates form what's called a motor unit. Antagonistic muscles are pairs of muscles that work opposite each other to produce smooth, controlled movement. Here's the rule. A muscle can only pull, it cannot push. That means if one muscle bends a joint, you need another to straighten it again. The muscle that contracts to create a movement is called the agonist. The muscle that relaxes to allow the movement or contracts to reverse it is the antagonist. Think about your elbow joint. When you bend your arm, the biceps contract. They're the agonist. At the same time, your triceps relax. When you want to extend the arm again, the roles reverse. The triceps contract and the biceps relax. These pairs are everywhere in the body. Elbow joint, biceps and triceps. Knee joint, quadriceps in the front of the thigh extend to the knee, while hamstrings in the back flex it. Ankle joint, calf muscles point the toes downward, while the tibialis anterior pulls the foot upward. Even in your face, the orbicularis oculi closes your eyes while the levator palpebrae opens them. The system ensures balance. If both muscles contracted fully at the same time, you'd lock up like a statue. If neither contracted, you'd collapse. Smooth movement comes from the precise coordination between agonist and antagonist. Skeletal muscles aren't uniform. They're made of two main types of fibers, slow twitch fibers and fast twitch fibers. Slow twitch, built for endurance. Fast twitch, built for power and speed. Both rely on the same basic sliding filament mechanism. Actin and myosin sliding past one another, but their internal chemistry makes them very different. Slow twitch fibers are like the marathon runners of your muscle system. They contract more slowly, but once they're going, they can sustain activity for a long time. They're packed with mitochondria, the cell's powerhouses, and rely on aerobic metabolism. That means they use oxygen efficiently to produce energy. They're also rich in myoglobin, a protein that stores oxygen, which gives them a darker reddish color. Because of this, slow twitch fibers resist fatigue. Perfect for posture, walking long distance, or endurance sports. Think of slow twitch fibers as hybrid cars. They're not flashy off the line, but they can go and go and go, sipping fuel efficiently the whole way. Fast twitch fibers are the sprinters. They contract quickly and generate lots of force, but they fatigue rapidly. They rely more on anaerobic metabolism, burning glucose without oxygen. This is fast but inefficient, producing lactic acid as a byproduct. They have fewer mitochondria and less myoglobin, which makes them pale in color, sometimes called white fibers. These fibers power explosive movements, sprinting, jumping, weightlifting, or throwing. Fast twitch fibers are like sports cars. They accelerate fast and hit high speeds, but they burn through fuel quickly and need lots of pit stops. What are proprioceptors? They're specialized sensory receptors found in muscles, tendons, and joints. They constantly send information back to your brain and spinal cord about muscle length, stretch, and tension. You already know the 5 classic senses: sight, hearing, touch, taste, smell, but proprioception is sometimes called the sixth sense because it tells you about your own body's position. Thanks to proprioceptors, you can touch your nose with your eyes closed, walk without staring at your feet, and adjust your grip when holding something heavy. Try this. Close your eyes and raise your right arm above your head. You still know exactly where it is because proprioceptors are talking to your brain nonstop. One key proprioceptor is the muscle spindle. Muscle spindles are embedded within the muscle fibers themselves. They detect changes in muscle length, basically how much a muscle is being stretched. When a muscle is stretched too far or too quickly, spindles send signals to the spinal cord, which triggers a reflex contraction to resist the stretch. This is the classic stretch reflex. When your doctor taps your knee with a reflex hammer, the sudden stretch activates muscle spindles in the quadriceps. They signal the spinal cord, and in response, your leg kicks out. Another essential proprioceptor lives in the tendons. It's the Golgi tendon organ. These receptors measure muscle tension, how much force the muscle is generating. If tension gets too high, they send an inhibitory signal to the spinal cord, causing the muscle to relax. This prevents excessive force that could damage tendons or bones. Think of the Golgi tendon organ as a safety valve. If you try to lift something far too heavy, the Golgi tendon organ steps in and tells your muscles, stop, you'll hurt yourself. This hidden sensory system, proprioception, is critical for everyday activities. When you walk on the uneven ground, proprioceptors adjust your posture step by step. When you catch a ball, proprioceptors fine-tune muscle force so you don't crush it or let it slip away. In athletes, training sharpens proprioceptive control. A gymnast landing a flip or a basketball player shooting a free throw relies heavily on this feedback. In rehab, exercises often target proprioception, retraining the system after injury or stroke. Voluntary movements are those you consciously control. They require planning, initiation, and coordination across multiple brain regions. They're goal-directed. They involve the motor cortex. Planning involves the premotor cortex and supplementary motor area. And the basal ganglia select the appropriate motor program, while the cerebellum fine-tunes timing and coordination. Examples of voluntary movements are deciding to stand up and walk across the room, writing your name on a piece of paper, or playing a piano scale. Imagine voluntary movement as typing an email. You consciously choose the words, sequence them, and then press send. In contrast, involuntary movements are automatic, often reflexive, and don't require conscious intent. They're stimulus-driven. They're mediated largely by the spinal cord, brainstem, and ANS. They can be protective or regulatory. Examples are tapping the knee tendon makes your leg kick without you thinking. That's called the patellar reflex. Blinking when something approaches your eye, and breathing and digestion, which are managed by automatic control. Involuntary movements is like your phone's background apps. They run automatically to keep the system going, even if you're not paying attention. Sequences of behavior are organized sets of actions performed in a specific order to achieve a goal. Instead of controlling each muscle contraction individually, The brain chunks actions together into a routine. Some examples are tying your shoelaces, driving a car and shifting gears, playing a song on the piano. Each of these behaviors involve dozens or even hundreds of movements, but we experience them as a single smooth sequence. Think of it like typing a word. You don't think about each keystroke, you think of the whole word, and your fingers run the sequence automatically. Several brain regions work together to produce sequences of behavior. The premotor cortex helps plan the sequence. The supplementary motor area is especially important for well-learned sequences. The basal ganglia plays a central role in selecting and initiating sequences, and the cerebellum fine-tunes timing and transition. Imagine a conductor leading an orchestra. Each section, strings, brass, percussion, knows those parts. But the conductor ensures everyone enters and exits at the right time. That's how your SMA and cerebellum manage your behavioral sequences. One of the fascinating things about behavioral sequence is how they move from conscious effort to automatic. At first, a new sequence feels clumsy. Learning to drive or dance takes a lot of conscious attention. With repetition, the sequence becomes smoother and faster. Neural circuits in the SMA and basal ganglia take over, freeing your conscious mind to focus on other things. Eventually, the behavior becomes almost automatic, like walking or brushing your teeth. Think back to when you first learned to type. You had to hunt for each other. Now your fingers fly across keys without you thinking about it. That's a behavioral sequence becoming automatic. Sequences of behavior are at the heart of everyday life. They let us multitask, build skills, and interact with the world smoothly. Athletes rely on well-trained sequences to execute plays. Musicians string notes into flowing melodies. Even language itself is a sequence, producing words and sentences in the right order. The beauty of the nervous system is its ability to reduce complexity, turning hundreds of micro movements into one unified action. At the heart of voluntary movement lies the primary motor cortex, located in the precentral gyrus of the frontal lobe. It's organized in a somatotopic map, often called the motor homunculus. Larger areas are devoted to parts of the body requiring fine control, like the hands and face, while areas needing less precision, like the back, take up less space. The motor cortex sends its signals down through the corticospinal tracts, which cross over in the medulla. That's why the left hemisphere controls the right, and vice versa. But the motor cortex isn't acting alone. Before you even lift a finger, the premotor cortex and supplementary motor area have already planned the sequence, considering context, posture, and timing. Think of the motor cortex as the conductor raising the baton. The score is written in advance by the premotor and supplementary motor areas, and once the signal starts, the orchestra of muscles plays in harmony. The cerebral cortex is about 6 layers thick and covers the surface of the forebrain. Within this sheet are specialized areas for movement. Primary motor cortex is located in the precentral gyrus of the frontal lobe. It sends direct commands to the spinal cord via the corticospinal tracts. Each area corresponds to a body part mapped in the famous motor homunculus. More space is given to parts needing fine control, like hands and face. Premotor cortex, located just anterior to the primary, it prepares movements in response to external cues. For example, reaching for your coffee cup when you see it on the table. The SMA, or supplementary motor area, is also anterior to the primary, but more medial. It helps plan and coordinate well-learned sequences like playing a piano scale or performing a dance routine. Together, these areas are sometimes called the motor strip, but they're much more than a strip. They're an interconnected network planning and execution action. Why inhibition of movements matters. Without inhibition, our bodies would be in constant motion, acting on every impulse. Inhibition is what allows us to stop actions that are inappropriate, switch smoothly between tasks, prevent conflicting movements from happening at once, and control reflexes when necessary. This ability is central to self-control, attention, and adaptive behavior. In fact, many movement disorders like Parkinson's or Huntington's disease can be understood as problems of too much or too little inhibition. Mirror neurons were first discovered in the 1990s by researchers studying monkeys in Parma, Italy. The scientists were recording neurons in the premotor cortex when they noticed something unexpected. First, a monkey's neurons fired when it picked up a peanut. Secondly, but the same neurons also fired when the monkey simply watched the researcher pick up the peanut. It was as though the monkey's brain was internally simulating the action it observed. This led to the term mirror neurons because the neurons mirrored the observed behavior. Scientists believe that mirror neurons play a significant role in action understanding. By simulating the observed action, we can understand its purpose. By imitating and learning, mirror neurons may underlie our ability to copy behaviors, which is critical in childhood development, and with empathy and social cognition. When you see someone smile or frown, your mirror system activates as if you were making the expression. It's like your brain is running a silent rehearsal of other people's actions, preparing you to join in, understand, and connect with them. The most famous pathways are the corticospinal tracts. There are fibers that run from the cerebral cortex down into the spinal cord. There are two main divisions. Lateral corticospinal tract originates mostly in the primary motor cortex, crosses over at the pyramids of the medulla, controls fine, precise movements of the hands, fingers, and toes. Examples are writing, playing a piano, threading a needle. The medial corticospinal tract travels on both sides of the spinal cord, not just one. It controls posture and trunk muscles, coordinating movements of the neck, shoulders, and torso. Examples are standing upright, bending to tie your shoes, keeping balance as you walk. Think of the lateral tract as your brain's scalpel for delicate, precise work, and the medial tract as its anchor, stabilizing your body while you move. The cerebellum sits at the back of the brain beneath the occipital lobes and behind the brainstem. It's divided into two hemispheres, connected by a midline structure called the vermis. Its surface is deeply folded, giving it a dense, compact look. Despite being only about 10% of the brain's weight, it folds over half of all of the neurons in the brain, mostly in the form of tiny granule cells. This incredible density is part of what allows the cerebellum to process information so quickly and precisely. Cerebellum is thought of as the coordinator of movement. Its role goes beyond simple coordination. Key functions include timing and rhythm, error correction, balance and posture, motor learning, cognitive contributions. Think of the cerebellum as your brain's quality control department. The motor cortex may design the blueprint, but the cerebellum checks for mistakes, fixes timing issues, and ensures the final product runs smoothly. Let's meet the major players in the cerebral microcircuit. Purkinje cells are the stars of the show. They're large branching neurons with flat, fan-like dendritic trees. They line up in a single layer and receive input from thousands of parallel fibers. They send their output in the deep cerebellar nuclei, which are the main output structures of the cerebellum. They're inhibitory, and they use GABA to regulate activity. Granule cells are the most numerous neurons in the entire brain. They're tiny, but they play a big role. Each one sends axons upward into the molecular layer where they split into parallel fibers. They run horizontally and synapse onto thousands of Purkinje cells. Climbing fibers are inputs that come from the inferior olive in the medulla. Each climbing fiber forms powerful, direct connections with a single Purkinje cell wrapping around its dendrites and strongly influencing its activity. Mossy fibers are inputs from the spinal cord and brainstem that connect indirectly to Purkinje cells by exciting granule cells first. And lastly, interneurons, such as stellate and basket cells, which fine-tune the system by providing additional inhibition to Purkinje cells. The basal ganglia are a group of subcortical nuclei buried deep in the forebrain. The major components include caudate nucleus and putamen, globulus pallidus, subthalamic nucleus, and substantia nigra. Even though they sit beneath the cortex, they have strong loops connecting with the frontal lobes, motor cortex, and thalamus. This makes them key players in voluntary movement. The basal ganglia don't send axons directly to the spinal cord. Instead, they work by regulating activity in the motor cortex via the thalamus. Here's the main idea. The direct pathway facilitates movement. It's the go signal, allowing a motor plan to proceed. The indirect pathway inhibits competing movements. It's the stop signal, suppressing unwanted actions. Dopamine from the substantia nigra pars compacta fine-tunes this balance, boosting the direct pathways and dampening the indirect one. This system ensures you don't activate every possible mode or program at once. Instead, the basal ganglia select the best one for the situation. Think of the basal ganglia as the bouncer at a nightclub. Only the right people get in, the right movements, while others are kept out. Too strict a bouncer and nobody gets in. Too lax and chaos breaks out. Perhaps the most famous movement disorder is Parkinson's disease. It results from degeneration of dopamine neurons in the substantia nigra pars compacta, which normally excite the direct pathway of the basal ganglia. Without this dopamine, initiating movement becomes extremely difficult. Symptoms include bradykinesia, which is slowness of movement, rigidity, stiffness of muscles, resting tremor, shaking that diminishes during voluntary motion, posture instability, difficulty maintaining balance. Treatments include L-DOPA, a precursor of dopamine that can temporarily restore function, and deep brain stimulation of the basal ganglia, which can help reduce symptoms. Parkinson's disease illustrates how vital dopamine is in balancing facilitation and inhibition of movement. Another classic condition is Huntington disease, a genetic disorder caused by a dominant mutation on chromosome 4. It leads to progressive degeneration of the caudate nucleus and putamen in the basal ganglia. Unlike Parkinson's, which causes too much inhibition, Huntington's causes too little inhibition. Symptoms include chorea, wild, uncontrolled jerky movements, impaired coordination, cognitive decline, and personality changes as the disease progresses. Huntington's shows what happens when the brain loses the ability to suppress unwanted movements. Movement disorders are not simply about bad muscles. They are about breakdowns in the circuits that plan, initiate, fine-tune, and execute movement. From the dopamine loss of Parkinson's to the genetic degeneration in Huntington's to the coordination problems of cerebellar ataxia, each disorder teaches us more about how the healthy brain works. Studying what goes wrong helps us appreciate the incredible complexity behind every smooth step, every graceful gesture, and every learned skill. Not all movement disorders are inherited, but genetics play a key role in several. Huntington's disease caused by a single dominant gene on chromosome 4. If a parent carries it, each child has a 50% chance of inheriting that gene. Parkinson's disease, most cases are sporadic, but some involve mutations in genes like alpha-synuclein or LRRK2. Dystonia and Tourette's can show familial patterns, though inheritance is often more complex. Heredity means that risk isn't just an individual issue, it extends to families, generations, and difficult decisions about the future. In healthy individuals, the CAG repeat count is usually between 10 and 35. If the repeat length stays in this range, the Huntington protein functions normally. In Huntington's disease, the number of repeats expands to 40 or more. The more repeats, the earlier the disease tends to appear. This is called anticipation. Imagine copying and pasting a word too many times in a sentence. Eventually, the message becomes unreadable. The same thing happens in DNA. Too many CAG repeats disrupt the protein's function. Genetic tests are now able to count the number of CAG repeats. A positive result gives certainty. People may struggle with anxiety, survivor's guilt, or tough choices about having children. Genetic counseling is crucial to help individuals and families navigate these decisions. The CAG test is both powerful and heavy. It can predict the future, but it can't change it. So here's the takeaway. Huntington's disease is caused by an expansion of CAG repeats in the HTT gene. More repeats mean earlier and more severe disease. This tiny genetic change disrupts the basal ganglia, leading to uncontrollable movements and cognitive decline. It's one of the clearest examples of how a repeating DNA sequence can shape the course of a human life, raising not just biological questions, but also ethical ones about testing, choice, and the future. See you next time.